Hello, it's my pleasure to welcome Dr. Christian Schafmeister from Temple University. I've known Christian for a very long time, about 20 years. We got started in, or I got to know him in a grad program at UCSF Biophysics, and I used to spend a lot of time hanging around in the computer graphics room, and Christian was in there as well. And I learned a lot, and I was always completely blown away because he was the first person I met who had designed a protein from scratch, which I thought was pretty amazing because I thought that that was a really hard problem, and he made it look really easy by writing a few computer programs, spending a few thousand hours characterizing it as well. Christian went on to do other great things, and now he's going to talk about a system he's built, which I think is really interesting because it's a combination of software engineering and science, and using the software engineering to push the science together forward faster than it would normally be. And I'm really excited about this because I've been using Christian's software for 20 years, and in fact, I just used his software a few years ago as well. And so it's got incredible longevity because I was using a 20-year-old program, and it's the state of the art. So I don't know whether that's a good thing or a bad thing, but he's going to tell us about what he's built more recently now.
Thank you. Thanks. Okay, so I'm going to tell you about the work that I've been doing over the last, 15 years, pretty much. I just wanted to start with the side of my group. So this is my group here. All of these fresh-faced young people there are synthetic chemists. They go into the lab every day and they make molecules. None of them do any software engineering or anything. I'm the only person who does that, and I do it in my copious spare time. I'm going to tell you about what we've been doing with molecules and the software and how I'm trying to bring this all together. Okay, so what I want to do is I want to build molecules the way that I can write code. I want to build large, complex molecular devices that can fix every problem that we have, that can build new materials, break down the old materials that we don't need anymore, like plastics, can convert carbon dioxide, sunlight, and water into fuel, basically fix everything. And it's all possible. We just have to figure out how to build the right kind of molecules. And this idea was basically first sort of articulated by Richard Fineman, who gave this brilliant lecture in 1959 that he called There's Plenty of Room at the Bottom. And in this talk, he basically proposed for the first time the idea of doing manufacturing, atomically precise manufacturing, on the molecular scale, and that if you could build devices down there, then we could build devices that are exactly the same from one to the next. And this is something that's really inspired me and what's driven me forward. All right, so there's already examples of this stuff. We're all made out of it. The main ones are called proteins. Proteins are long chains of amino acids. They're like charm bracelets. And when you put them in water, they fold up into well-defined three-dimensional shapes that do things like speed up chemical reactions, trillions of fold. They build materials, they build our muscles. Everything that we're made out of was built with these machines. They can do things like organize atoms in order to speed up chemical reactions inside of little pockets. Antibodies are big molecules that have these two binding sites up on the top left and right there. They can pretty much recognize and bind almost any other protein, and so they protect us from viruses. If we didn't have these things, we wouldn't last a few days. We would die. They make channels in membranes that pass ions and water and sugars and all sorts of things in and out of cells, and they're perfect filters. There are amazing capabilities that are waiting for us to develop if we could build things like this. Now, I originally thought the way to do this was to build things like that, to build proteins. And I spent 4 years building this thing. This is the protein that David just talked about. It's 4-HB1. I spent 4 years putting this thing together. I wrote some software. I designed a gene. I expressed the protein. I purified it, crystallized it, showed that it was well folded. And the thing is a molecular boat anchor. It doesn't do anything. And the big problem is I didn't know how to make another one. And I didn't know how to make ones that are more complex and could do all those wonderful things that I showed you before. So I got very fed up with proteins at the end of my graduate career. This is another problem. I mean, they're amazing. They have amazing capabilities. You take some proteins, you hold them at 38 degrees for 35 days, and you can make things like eagles and people and build wonderful functional things. They're also very fragile. You hold them at 70 degrees for a few minutes, and you've got scrambled egg, which is delicious, but not very functional. So making proteins and using them for things that people want to use them for, like sensors and catalysts and stuff, they're just very, very fragile. We need better materials to build molecules that do things with. That's what I thought. Here's another problem. Computationally, they're almost intractable. Right? So this is a chain of 10 amino acids. It's a 10-mer peptide, a short little section of a protein. A real protein is usually 100 to 1,000 amino acids. But just 10 amino acids like this, strung together in a little charm bracelet, if you go through the math and you work out how many shapes can this molecule have, because every one of these little lines is a bond around which you have essentially free rotation. And if you just do some simple arithmetic and you ask, well, how many shapes can this molecule adopt? Very conservatively, it's about 2 times 10 to the 14th different shapes. So this is really not just one molecule. This is 2 times 10 to the 14th different molecules, because every shape can do different things, potentially. And it's constantly flickering and moving between them. That's a problem. Now, modern processors are not very fast. I mean, they're great for video games, right? Pew, pew, you can do amazing graphics and stuff with them. But for doing chemistry, they're pretty slow. You got about 10 to the 9th floating point operations per second on a modern CPU. If it takes you 10,000 flops to generate one of those conformations, it'll take you about 6 years per molecule on one CPU to generate every conformation of just this little molecule. All right, 6 years for one molecule. And then there's the question of how many of them are there. If you take all the 20 amino acids and you put them into every position, in every permutation, there are 10 to the 13th different molecules that you would want to search through in order to find one that does something like, mimic insulin or bind human growth hormone receptor. These are huge numbers, a huge problem for computational design of molecules. So what I thought was, well, folding is the big problem, this idea of the molecule undergoing all these different conformational shapes. So what if we eliminate it? What if instead of making building blocks that connect together through a single bond, we make building blocks that are rings and we connect them through pairs of bonds? So we make ladder molecules. And then we can hang our chemically reactive functional groups off of it. What would that give us then? Well, for one thing, it massively reduces the number of conformations, shapes that it can adopt. You're down to like a few thousand. All right, so 10 to the 10th fewer shapes that it can adopt. The amount of time that it takes to build every possible shape is now 0.06 seconds. All right, so it's 3 billion times less work to build all the shapes that this molecule can adopt. Pretty much it's a rod, and the side chains, those R groups that are hanging off, they flop around a little bit. And how many of these 10 mers are there? Well, if we use the same kind of side chains that are in proteins, you actually have about 1 million more molecules that you can build, because all those little stars in there, they represent what are called stereoisomers. It's basically one bit of information that defines the shape of the groups attached to the carbon that each of those stars is next to. So you can use this thing called stereochemistry to define the shape of the molecule and program shape in that way. So you still get a lot of diversity. You still have a big design problem. So this is what I've actually come up with in the last 15 years, and we've been developing it to the point that we're at now. So we make these building blocks. They are cyclic building blocks. They're like little Lego bricks. And they connect together. This building block connects to a next one through a pair of bonds at these two atoms. And these connect to another building block through another pair. So here's what this looks like in real chemistry. Building block is connected to another building block through a pair of bonds. So you get a ladder molecule. And we can hang side chains, groups, off of it. And you get a molecule that has a programmable shape depending on what stereocenters you put in there, what rings you put in there, where you put the different linking groups, the amino acids on the backbone. You've got a lot of choices here, a lot of design choices. And basically what we have are these little Lego bricks. You snap together through pairs of bonds. We can put them together in any sequence. And we can make these little sticks with stuff hanging off of them. Okay, the kind of things that we can do with this. Well, we could put, a couple, 2, 3, 4, 5 of these little sticks together like your fingers and make molecules that display a lot of surface area and could wrap around a protein. All right, all the medicines, if you take like blood pressure medication or aspirin or something, these are very, very small molecules that would be maybe about this size here. They could plug into a greasy pocket inside of an enzyme and jam it. That's what the pharmaceutical industry is based on. The biotech industry is based on new therapies based on antibodies, which are these big molecules that actually wrap around the protein. So what we can do here is use chemistry to make molecules that work like antibodies do, but are just maybe 5% of the size. So they could get into places where antibodies can't. They also would be indestructible. They wouldn't need a cold chain, a 4-degree, to be kept at 4 degrees all the time in order to maintain their viability. You'd have a lot less problems with like batch-to-batch variation because you basically grow these antibodies, and from batch to batch you can get different results. Okay, but they can vary just as much surface area when they bind a protein as an antibody does. This is the big thing I want to do. I want to make catalysts. I want to make molecules that can speed up chemical reactions. So I need to make big molecules that have pockets inside of them that I can organize multiple groups in order to push and pull on the electrons and change a molecule that goes into this pocket and another molecule come out. If we could develop this capability, we could solve every problem we have. Aging, right? You get all these cross-links that cause our skin to get cross-linked and start wrinkling as we get older. Well, you could think about designing small catalysts that are a few percent of the size of a regular enzyme that could reverse those cross-links. They could get in and just loosen everything up so your natural enzymes can get in and start fixing things. There's all sorts of ideas that you can think about to if you could quickly develop catalysts the way we write software. Okay, and what it's all about is about organizing groups in three-dimensional space with the right constellation and finding a scaffold behind it that can hold them in place. We could develop new ways of killing bacteria. So we can make these triangular pores that could bury themselves in bacterial membranes, hold open a hole, cause a bacteria to bleed its guts out. I'm just throwing up some ideas of what we can do with this kind of shape control. By sticking greasy groups into the center of these triangular molecules that I'm proposing here, we can make receptors that could bind almost any small molecule and be used to detect small molecules like nerve agents, for instance, or other agents. We can also start to build channels. We can make molecules that could selectively pass water through them and make perfect sort of water filtration membranes for reverse osmosis, for desalinization, or separate ions from seawater. If I could, pull gold out of seawater, I wouldn't need to write grant proposals anymore. So those are just some ideas. Let me tell you about some of the things that we've actually done with these in the last 15 years. So this is a molecule here up on the upper right that we designed to bind a protein called MDM2. It presents these 3 green groups, this one, and this one, and it organizes them in just the right constellation so that they can slot into this groove on this protein called MDM2, which is actually a protein that's involved in a lot of cancers. And we designed this molecule using the old version of my software. And when we threw it at the protein, it stuck fairly weakly, but it did stick to the protein. Subsequently, we've improved the molecule, and we've been able to make it bind reasonably well. And when we throw it at cells, what we find is it gets into the cells by passive diffusion. It has a green fluorescent group on it, so it goes into the cells and becomes fluorescent. These are confocal microscope images of cells, and you can see the compound is all throughout the cell. And it gets in and it stabilizes the protein that we designed it to bind. The levels of that protein shoot up about 30-fold. And there's another molecule that Roche was actually developing to be anti-cancer agent. It binds the exact same pocket on the protein and kicks our molecule out, when we mix them together, so it demonstrates that they're binding the same pocket. It doesn't do this. So our molecules, I think because they're big and structured, they do things that small molecules don't do. We published this a couple of years ago. We've now been able to, by changing the groups that hang off of the scaffold, we've been able to improve the affinity about 10-fold. And also when we change the groups that we think will not bind the protein, we completely wipe out the binding. So you have to present the right groups and present them in the right constellation, and then you get this binding activity. Another thing we've done is we've made catalysts. We make molecules that speed up chemical reactions. So this is the same kind of scaffold, right? All these building blocks connected through pairs of bonds. Here they're displaying a base, an alcohol, and a urea. And because they can arrange all of these groups in the right constellation, you can throw in methanol and vinyl trifluoroacetate, and this molecule will transform into a new product, the methyl trifluoroacetate there. And what we can see here is product, the amount of product with time. You get a buildup of product until you use up all the starting materials. And if we make any change in this molecule, we change the backbone geometry or how the side chains are presented, we lose activity. Right? So we've got the ability to sort of design and create a somewhat optimal molecule for this chemical reaction. This is what the thing looks like when you model it. Here's the sort of ladder molecule backbone, and it can present the base, the alcohol, and the urea in this constellation, so that when the reagent comes in here, it can get attacked by this oxygen and activated by this carbonyl, and this alcohol here gets its proton pulled off by this base here. So it's really a device. It is organizing the groups in order to carry out this specific function. But what I really want to do is I want to build big molecules. These things that we've made over the last couple of years have been just basically one of these segments, and we've developed a couple of demonstrations of applications for those. But they're still small molecules. They're about 700 Daltons, 800 Daltons, which is, you know, like a drug-sized molecule. And I don't think they're any better than anybody else's small molecules. They're just versatile. What I want to do is I want to string 3, 4, 5 of them together to create molecules with pockets and large surfaces. Then we can really start to do new things, because organic chemists can't make complex structured molecules of this size. We're the only people who can do that with this kind of approach. Got to be careful there, but in terms of making asymmetric, complex, programmable molecules, I really think we've got the best and only way outside of proteins and DNA to do that. So that's where the sort of software comes in. I've got a design problem. I want to build these big structured molecules, and ultimately I want to connect them together to create even larger things like robots that can go into the body and fix things. That's going to take a lot of work. We're going to have to get really good at designing these molecules and connecting them together. Okay, so I've got this molecular Lego, this instruction set for functional molecules. I'm going to put this away here. And I think they can do all the sorts of things that proteins can do, we can synthesize a lot of them, but I have a design problem. I have to pick out of this enormous universe of molecules that I can make the ones that could actually do something useful. Okay, so to do this, I basically need an oracle. I need something where I can say, I have a certain requirements for a functional molecule. I want to organize these 4 functional groups in just the right constellation, or around a pocket. And I want this oracle to tell me what's the sequence of the molecules that we can then assemble in the lab that could organize those groups that way. That's what the problem comes down to. A 3-dimensional arrangement of groups held in space. What's the sequence of one of our molecules that could hold those groups in space? So I've been writing software that could let me do this. Okay, now the question is, how do we design molecules with computers? There's a couple of ways you could do it. One idea would be to just take some subset of the sort of 10 to the 160th medium-sized molecules that chemi-informatics people tell us you can build that are possible. Take some subset of those and then just build 3-dimensional structures for them and ram them through a series of computational filters and come out at the bottom with a few molecules that could satisfy your requirements, and then go and try and convince a chemist to make that molecule. Spend a year or 2 of their lives trying to make that molecule. We do this for small molecules. That's how all the things that I showed you so far were sort of designed. One advantage that we have, though, is that our library of molecules that we start with can all be made with exactly the same synthesis. right? So we make molecules, and so we only design molecules based on the scaffold that we can synthesize in the lab. A more powerful way of searching through sequence and structure or conformational functional space is to use Monte Carlo, where basically you pick a random starting structure, and then you mutate it, and you score it based on how well it satisfies your criterion. And I'm saying here, in the center where it's light, that's where optimal solutions are. And out here are suboptimal. And you just mutate, check, and occasionally your mutation makes things worse. You roll a dice based on a temperature factor, and you decide whether to keep that or not. And you basically just move through space trying to find a locally optimal sequence. Right? So that's Monte Carlo. Now, the software requirements to do these two things are a little bit different. So screening, you can basically use off-the-shelf software, and you can run it on separate processors, and you've got some serialization format that you can write your data out. You can send the data between one processor and the next, and you can do pretty efficient screening that way. The Monte Carlo optimization molecules requires different kind of software. It requires basically the ability to build the three-dimensional structure, score the three-dimensional structure, and then mutate the molecule. It requires them all to be tightly coupled together in one processor. Because if you're sending stuff between processors, you're wasting a lot of time serializing and deserializing and stuff like that. So I'm convinced that to do the Monte Carlo optimization on the right, it needs a programming language that's really dedicated to this. You need something that can generate code that runs as fast as possible to do this kind of Monte Carlo optimization of molecular structure. So I went off on a little side project. I decided to try and implement this. I spent 2 years working with Python and Boost Python and all this 100,000 plus lines of C++ code that I had written to build molecules. And I basically almost went nuts because I just can't control every aspect of this thing. Python developers made certain choices about the way memory management is done. C++ is done a different way, and getting everything to talk to each other. Everything that I do, I basically have to think about memory management and just all sorts of stuff like that. And it just really drove me crazy. I wanted a computing environment where I could control every aspect of it. Mostly what I wanted is a computing environment where all my atoms and bonds and everything would be garbage collected. I wouldn't have to worry about memory management. So I took the C++ code that I wrote, and C++ is an excellent language for building compact arrays of complex data, and writing cache-aware code is excellent for that. Writing more complex software where you want to try something one day and then try something else another day, there it's not quite as good because I always felt like I kind of got locked in on the code. So I just threw together a sort of archaic interpreted Lisp interpreter within CANDO. It could do if and looping and things like that. And all the molecules that I just showed you, I designed using that software. And I ran this software with this archaic Lisp running on about 40,000 CPU supercomputers in what was then the TeraGrid. And it designed those molecules, and it was wonderful. Because my molecules and stuff were all garbage collected, I didn't have to worry about a lot of software development issues. So I liked that, but I didn't want to spin off into the ether developing a whole new language for chemistry. I didn't think anybody would use it, and I'd have to document it, and I had all these issues about that. So I decided to use a Lisp that lots of people were already familiar with, and I had a choice of Scheme or Common Lisp, and I decided on Common Lisp because it just seemed a lot more full-featured for what I wanted to do. So I wrote a new compiler for Common Lisp. It's called Clasp. It stands for Common Lisp in Scientific Programming. And it is the foundation that the CANDO software, the molecular design environment that I've written is built on. And basically, I had to write a new Common Lisp because I want Common Lisp to talk to C++. And there are very few languages that can interoperate with C++ in the way that I needed them to interoperate. So basically what this compiler does is it takes Common Lisp code at the top and C++ code. It generates LLVMIR. At the time that I was starting this, LLVM was becoming a thing, and it is a wonderful thing. It's sort of a common language for sort of portable machine language where I don't have to worry about the details of the processors underneath. I can just write LLVMIR, and it gives me the ability to interface to other LLVM languages. And there are really smart people working on turning this stuff into very fast native code for different processors. So that's what I did. Now, why choose Common Lisp, especially for a scientific language? It's not very commonly used. Well, it's a compiled language that has a lot of high-level language features that just haven't made it into other languages. I mean, I'll mention some of them, but it's got things like macros, where you can write programs that write programs. And that's very powerful. It's got a great condition system. It's just got a lot of stuff that just hasn't made it into other languages yet. It's got a well-defined specification that the Department of Defense funded back in the early 90s. It's got package managers, ASDF and QuickLisp, for bringing other people's Common Lisp code into your environment. And they're all written in Common Lisp. It's a self-hosted language, and thus, if I wrote a compiler in Common Lisp, I would have access to it, and I could tinker with it to make it generate things like OpenCL, or to generate specialized code that has to run really fast. There's a bunch of existing implementations for it. There's a great user community and an ecosystem of existing software. It's also a tried and true industrial strength language. It's used to run Google Flight products. You guys use that. And there's a bunch of other companies that use Common Lisp. So the main thing about using it for CANDO is it lets me garbage collect all my chemistry objects. So atoms and bonds, or they're implemented in C++, they're exposed to Common Lisp, and they're all garbage collected. And I have compacting garbage collection working with C++ objects. I'll tell you a little bit about that. It's got true macros. So macros are functions that can generate code on the fly. All right, it's what C++ template programming is mimicking. But I don't know, this got a good laugh at Cambridge. I don't know about here, but basically the way I think about C++ macros and C++ template programming and Common Lisp macros are C++ template programming are to Common Lisp macros what IRS tax forms are to poetry. Because C++ template programming, it lets you write a chunk of code where you can sort of slot in types. Okay, and that's very powerful. I do a lot of template programming. Okay, I like it. It's powerful. It's difficult, though, to convey your intention to another programmer because it's sort of really spread out code. Macros, Common Lisp macros, let you generate code on the fly in a much more natural way. It's got dynamic variables. So dynamic variables are like globals, but you can set them, and then all the code that you call underneath that uses those variables. But as soon as you return, the old values get restored. So for chemistry, it's great because what it would let you do is configure global variables, run some code, and then configure the global variables again a different way, and then run that code again. And it lets you, it really facilitates writing software that can interoperate with other software in the same ecosystem. Emacs is a great example of this. Emacs is, thousands of people have contributed to Emacs, and what I think really makes that work is dynamic variables. Generic functions. I can write functions that dispatch not on just one class, but multiple classes of multiple arguments. So, often I'll have, I want a function where I want to call it with a particular energy function and a force field. That's, I realize I'm spinning a little bit out of stuff you guys are, work with every day. There's a bunch of other features in Common Lisp that are great for chemistry, and I'd love to go into more detail about that with anybody who asks. So Clasp is about 150,000 lines of C++ code. There's about 30,000 lines of Common Lisp code that I stole from another implementation called ECL. And it's also a C++ template library that lets me easily expose C++ libraries into the Common Lisp environment. So it's sort of like Boost Python. I can just give it the name of a function in Common Lisp, a pointer to the function in C++, and at the C++ compile time, it generates a wrapper function for it. And so it makes it very easy to expose C++ libraries to Clasp. Using CLBind, I expose the LLVM API, the C++ API. So I am using LLVM C++ API and expose it to Common Lisp. I also exposed a bunch of the Clang API, namely the AST, Clang AST library and the AST matcher library. I do that because I need to sort of grovel through my C++ code to build an interface to my garbage collector. And I'll tell you a little bit about that. I've implemented tag pointers for fixed nums, characters, and single floats, so that lets you generate really fast code. And Clasp supports ASDF, Slime, and Quick Lisp. Those are Lisp things written in Common Lisp that Clasp uses. So basically what it is, all you need is a C++ compiler Clang to compile this thing. It starts up an interpreter that's really slow. That loads some Common Lisp code, which is a compiler. It compiles itself, gets quite a bit faster. And then the bootstrapping compiler compiles another compiler that I got from my collaborator, Robert Strand, called Cleaver. And Cleaver is going to be a state-of-the-art Common Lisp compiler. It will generate performant code, and I can show you something in a second. Clasp is very similar to ECL, the embedded Common Lisp, which is a Common Lisp based on C. And I've done that on purpose so that I can use a lot of their code. And Clasp also does just-in-time compilation and ahead-of-time compilation. Okay, so Cleaver is a compiler developed by Robert Strand here. He's a professor of computer science at University of Bordeaux. He's got decades of experience writing compilers. And he's trying to write a Common Lisp compiler that's written using all the best features of Common Lisp. And it is a wonderful compiler. I've spent the last couple of months hooking it into mine, and I can now generate code with it that runs about 4 times as fast as C now, will soon be, I think, comparable. So this is Robert here, this is me. We both get our hair styled by the same person, good old Mother Nature. And yeah, he's developing this in this project called Cycle, which is there on GitHub. And what Cleaver does is it takes S expressions, their Common Lisp code, generates these intermediate representations here, which shows sort of the flow of processing and the flow of data. And then my Clasp code takes this and turns it into LLVMIR, and then some of you guys help me get that down to native code. Yeah, there's a couple of other things. I generate dwarf debugging information, so I can use LLVB to debug Common Lisp and C++ together. My stack frames are interleaved C++, Common Lisp, C++, Common Lisp. Clasp supports 2 garbage collectors. It's got the main one is going to be the memory pool system, which is a compacting garbage collector, which means it moves C++ objects around in memory, constantly compacts them, so it could run essentially forever on a fixed footprint of memory. Right? I use the Boehm GC just to bootstrap this GC. So it's the first Common Lisp built on LLVM. Yeah, I think that's about all I want to say here. Okay, so exposing C++ libraries to Clasp. We've got a C++ function here that takes 2 arguments, returns another argument. All you have to do to expose it to the Common Lisp is write def, add numbers, give it the string here, that is the name in Common Lisp, a pointer to the function, and then CLBind, the wrapper C++ template library, which I stole from LuaBind, which was built on Boost Python, will build a wrapper function for it that converts the types from Common Lisp to C++, calls the C++ function, and then takes the return value and converts it back again. And that's a very fast round trip because everything is C++ underneath. So this is something I've just generated in the last couple of days. It just sort of shows what the performance is as of a couple of days ago. This is sort of the tip of the spear in terms of performance. What I'm doing is I've set up inlining in the Cleaver compiler. Robert Strand has helped me, and we've gotten inlining working. And just a simple little algorithm that calculates the 78th Fibonacci number 100 million times, the C-class compiler will run at about 4 times slower than C and Steelbank Common Lisp, which are comparable. My original compiler that bootstraps the system is considerably slower. So just in the last couple of months, I've been able to speed this thing up about 220 times, and I'm now very close to performant code. What's missing is type inference, and Robert is building that in. Once we have type inference, I can see all these repeated type checks will be stripped out of the code, and it should run very close to sort of C++ speeds. Okay, so then there's CANDO. So I'm going to, let me see. Going over here. I have till like 10:02, right? Yeah. Okay, so just 10 more minutes. I'll give you a quick tour. So CANDO is the molecular design environment that runs within Clasp, and that runs on top of C++ and LLVM. CANDO can do all sorts of chemistry. I'm a chemistry professor. I teach undergraduates. I crush the dreams of undergraduates who want to become medical students because they're not studying hard enough. But I teach a lot of chemistry, organic chemistry, and so I've built a lot of this stuff into CANDO. So it lets you build and represent molecular structures. I can build force fields. I've got Amber built into it. It's got energy terms for things like chiral restraints. It's important to maintain the sort of stereochemical configuration around carbons, so I've built a restraint term in there. It's got a very fast energy minimizer that uses 3 separate algorithms: steepest descent, conjugate gradients, and then a truncated Newton-Raphson method. And it switches between the different algorithms as you get closer to the solution. So it very quickly converges on good geometry. I've got automatic type assignment built in there. It knows canonical prelog rules, something I wish I could teach all my undergraduates. It's got smart chemical pattern recognition, which is like regular expressions for molecular fragments built into it. Because it's got a whole language, Common Lisp built into it, you can rapidly build complex chemistry tools within it. And everything's garbage collected internally. So you don't have to worry about memory management. I can hook in all this external software as well. I've hooked in LLVM and Clang. The same way I can hook in things like OpenMM to do molecular dynamics, or Amber, or OpenBabel. OpenBabel is a software that will convert 100 different chemistry file formats into 100 other file formats. I get one of my collaborators on the internet is hooking in SGML, so we can get graphics and OpenGL graphics in there. We could do electronic structure calculations. We can hook all this code in and expose it to the Common Lisp environment, and then you could use it from within Clasp rather than running separate sort of standalone programs. So here's just an example of how I'm using this thing. Okay, so I want to develop molecules that can cure HIV. We've been working on this for 2 years. Trying to make a molecule that can prevent HIV viruses from fusing with human membranes. And what they do in order to fuse with human membranes, they've got this protein on them called GP120, GP41, which is on the surface of the viral coat. When it comes near a human cell, it recognizes something on that cell and sticks these greasy tails into the human cell. And then it undergoes this massive conformational change where it folds up and it pulls the 2 membranes together so they can fuse, and then the viral contents can get injected into the human cell. This is a proven mechanism that when we make molecules that jam this process, HIV can't infect human cells. And Roche developed a drug called Fusion that was available for a while. It might still be. There's a huge peptide, this huge peptide here, that mimics part of the fusion machinery. Okay, and the fusion machinery, here's a picture of it. This is the GP120 from the crystal structure. And here we're looking at it sort of top down. There are these 3 inner helices, and then there are 3 outer helices, this one's missing, that are part of this the final sort of fusion complex. Now the idea is, if you can make a molecule that can mimic one of these helices, that can bind down this groove, then you could prevent the machinery from folding over and you could prevent the viral fusion. All right, so Roche was synthesizing and selling this molecule, but it's a very difficult molecule to administer. You've got to basically inject 1 ml of a concentrated peptide solution into the muscle every day, because this stuff gets chewed up within minutes. It's a peptide. Our bodies know all about peptides and how to chew them up. Some people would rather die than inject themselves with something like that every day. It's a painful process. So really what you want are molecules that you could take as a pill, right, that would get across the gut and get in and jam this machinery. So that's what we're trying to develop. We're trying to develop a molecule that can replace one of these helices and basically stretch down this groove that would survive your gut, that would survive all the proteases in our bloodstream that would chew up this thing very quickly. And so we're designing that. And here's the top end of it. This is the top of the helix. You've got a tryptophan, isoleucine, and a aspartic acid here. Using this design software, we have designed a molecule based on our scaffold that can present 1, 2, 3, 4 groups on the scaffold with the right stereochemistry. And we just managed to synthesize and purify this like last week, and we're going to start throwing it at the protein to see if it sticks. So we don't know the answer to this. It's been 2 years we've been working on this. Chemistry is hard. It's much easier to write software. Okay, so then what we're going to do is if it does stick up here, if we can get a toehold, then we can continue to extend the molecule down, build more and more stuff off of it to make more contacts so that we can bind tightly to this groove and prevent the machinery from folding up. Because we've got to compete against this helix here, which is already there. It's part of the protein. It's like we got to put a molecule in there that's going to jam the closing of these scissors, basically, and that's a tough challenge. Okay, so here's how we do it. Draw the molecule in ChemDraw. ChemDraw is a program that lets you draw molecules that every chemist uses. So we sketch the backbone of the molecule, we annotate it. Here I'm just using the atom names, but there are other ways to annotate this molecule. I give it some information here. So it's a cartoon of a molecule with a bunch of annotations where I'm sort of hijacking ChemDraw's sort of drawing abilities to annotate the molecule. Then I load it into CANDO. You work with CANDO in Emacs using this programming environment called Slime. Well, basically, here I'm writing functions that are Common Lisp functions that build molecules. Here I'm giving instructions. I'm telling CANDO what to do with the functions and the molecules that it loads. And here is a window where CANDO is running and generates its output. Okay, this is a REPL, a read-eval print loop system. And basically, you just put the cursor on one of these lines, you hit a key, and it sends this expression to CANDO. It does something and generates output. So what are the kind of things that I do? Well, I want to read in AMBER type rules and parameters. So I'm reading the antechamber type rules up there. Once this executes, I've got a variable called *ff star. It's a dynamic variable that contains the entire AMBER force field and type rules in it. Then I can load that ChemDraw file I showed you. And once this executes, I've got this dynamic variable ag, which contains the annotated molecule that I drew in ChemDraw. Then I suck out the stereocenters, the carbons that I can configure, and there are 5 of them in this molecule that could be either S or R. So each one is like one bit of information that I can control. So there are 32 possible configurations of this molecule. And since I don't have any idea what the shape of this molecule is right now, I don't have a crystal structure or anything, I have to start from scratch. And scratch for me is just throw random coordinates at each of the atoms. And you get this massive tangled net. I send it to this program Chimera, which is developed up at UCSF to render molecules. And then I want a 3-dimensional structure. I just say, well, build good geometry from random coordinates. Okay, so give it the molecule I built, the force field. What it does is it automatically assigns types. It automatically assigns force field parameters and chiral restraints based on stereochemistry I told it. Then it minimizes the energy, and as it gets closer to the solution, it switches between these 3 different methods. Then it checks if the geometry is good, because they got a lot of rings. Often rings will sort of interpenetrate each other. You'll get rings that are chained, locked together, and that's not good, a good molecule. So it checks the geometry, and if it's bad, it goes back up to here, and it just keeps doing this. Okay, this is all implemented in Common Lisp on top of C++ code. And then I want to generate all stereoisomers. I just write a loop. It iterates through all 32 of them. It uses the integer value, 0 to 31, the binary pattern to represent S and R in each position. It assigns the stereochemistry, builds a geometry for the molecule, writes it out to a file in a mol2 format, and then I can say array them all in one structure, and there they all are, 32 molecules. And this takes about 15 minutes to generate it this way. Just to show you that these have different shapes, what I did is I took that molecule, I said, take 3 atoms from that top right corner there, and superimpose them for every one of these 32 structures. Here's 2 of them superimposed, so here's that thing at the top there superimposed, and you can see these are very different shapes. The blue versus the brown, very different shape. There's all 32 of them on top of each other. This is the superimposed stuff here, and you can see you've got 32 very different shaped molecules. All right, so these are the molecules that I can now use to screen and computationally look for the one that might bind that groove the best. Okay, so I've gone way down the rabbit hole here. I'm a chemist. I'm supposed to be writing grant proposals and guiding students to make molecules and stuff. But if I don't know what molecules to have them make, we're kind of stuck. We can make these complex, huge molecules, but I need a way to design them. It's either that or just make a whole bunch of them and just screen them, which has got its own problems, its own challenges. So this is what I've done, and I think I'm coming out the other side with something in my teeth that we can actually use to design molecules. And so that's basically what I've done. I've developed this hardware, the stereoisomers, these molecules that are sort of a new instruction set for building functional matter, software to help us design it, and we're moving forward, hopefully if funding allows, to try and develop molecules that can cure diseases, bind small molecules, act as channels, speed up chemical reactions, all sorts of things. So Clasp is available. It's on the internet. It's on GitHub. Runs on OS X and Linux. And CANDO will be as soon as I get the licensing stuff worked out with Temple. With that, thank you very much for inviting me, and I would love to answer any questions that you have. Yes?
So tell me more about the synthetic ability. Like, I mean, the Lego building blocks sound very appealing, but what can you actually do synthetically? Can you build one with 50 blocks together? Like, what are the limits of what you could do and how hard is it?
So the 50 is tough. We've made 8 MERS. 8 MERS are 4 nanometers long. There's not much you want to do with that.
And is that the same reaction? Like, do you know ahead of time, or do you have to, like, does a synthetic chemist have to think really hard about that?
I didn't put that stuff up, but no, you don't have to think about it hard at all. It's the same kind of chemistry that you can buy on the internet. DNA, you can buy DNA oligonucleotides. You can buy peptides. And it's basically robots can mix together chemicals one at a time. You start with a plastic resin that's got a group on it that you can attach something to. You throw in a lot of that something. It attaches. You take off the protecting group. Then you bring in the next one, and you attach it. And then you bring in the next one. Cycle time is about 2 hours. You can assemble these one building block at a time with precise control over the sequence because you're adding building block X, then Y, then Z.
So why do you stop at 8? Like, what's the limit? Why can't you make it?
I don't know what to do with anything of that length. Everything we do, it is difficult because we have to synthesize all of these building blocks ourselves, and we've turned that into process chemistry where industrial, you could make ton quantities of these building blocks. But right now we make them in the lab by hand, 150 grams at a time, and then we start using it. So there is a fair amount of effort expended, a lot of effort expended in that. Putting them together is fairly straightforward. But we've spent the last only 6 years ago, were we able to start putting a functional group on them. So we've been developing all of this stuff sort of as we go along. But you can assemble these things the same way you assemble companies robots assemble peptides. We do everything by hand right now because it's just more efficient to do things by hand. Get better product out of it. Yes, David.
It looked like you actually had a branch art group that had an extra backbone. If you go back one page, bottom right, are those art groups that have backbone on them, or is that a thing that attaches the rigid rods?
So each one of these represents one of these building blocks, and this thing represents the side chain. We can put chemical reactivity on there that can cross-link with other side chains. So we can create branched and networked cross-linked structures. There isn't a lot, there's a fair amount of sort of cross-linking chemistry that we can use to put things together. Yeah.
And then how do you maintain the rigid rod angles with respect to?
Oh, you just get that. That's for free. I mean, you're not going to be able to change the shape of this.
But two rods, how do you get their angles constant?
Oh, you'd need to make a ring. If you take two rods and you connect them through a little tether, they will flop around like logs that are chained together on one end. At 300 Kelvin, room temperature, things are, solvent is hammering into everything and they move around a lot. So we started to tie them together into rings called macrocycles, and we're just trying to get the first paper of that published. But we've made a bunch of these macrocycles. We haven't published any of them yet. And the trick there is to make the cross-linkers as short as possible so that you can still control the shapes. They don't roll around or flop around.
I would expect them to pucker.
They pucker, they flop, they do everything. The problem is not that these things are too rigid. The problem is that they're still too flexible. You've got to wrestle everything into position. And this is something I have a lot of trouble convincing my chemistry colleagues. They think that dynamics is really important, that these are like diamond. They are not. They're very flexible. They're just not crazy flexible like peptides and peptoids and DNA are. I'm sorry. Yeah.
Sorry.
No?
Oh,
Sure.
So you make these computational predictions about the molecules in design. How accurate do you find these predictions are in practice? Like, do you have, like, I know it's hard to put a number, but like 30, 50, 70?
In terms of the stereochemistry and the connectivity and stuff, it's perfect. I mean, we know exactly what we're putting together. We've gotten a couple of crystal structures. They look exactly like what you build. It's almost boring. They look exactly like what you build with a plastic model set. Now, these bigger things, we barely made a few of them, just enough to get mass spec, not enough to get structural determination. And I'm dying to see what they look like in reality. And if they have shape, trying to make something that has shape and then see what that shape is and see if it looks like what we predict it to look like.
It's like, 3D protein structure prediction is super, super hard. Is there like a chemical reason to believe why you made large molecules, liquids, why you'd be able to like predict the structure more accurately than you would for protein?
Because I can predict the structure of that. There's far fewer degrees of freedom here to worry about.
Because you can control the backfill more finely. You should be able to predict.
You will. I mean, I'm paying the cost of all that computational searching in the design and synthesis of these molecules. They're more computationally tractable. Yeah. I mean, it's easier to predict. It's easy to predict the shape of a ladder. It's not easy to predict the shape of a piece of string. A ladder has one shape. A piece of string has infinite number of shapes. That's the fundamental there. Yes.
Also, on your slide of Unitarian versus Monte Carlo.
Yeah.
Monte Carlo needs well-defined thickness over your whole space. Can you explain in layman terms what is that thickness?
So fitness for for binding a protein would be, I've got 3, 4, 5 functional groups. There's a bunch of fitness functions we can use. One is just, I've got these 3, 4, 5 functional groups arranged in a particular constellation in space, and can I make a scaffold that mimics that constellation? So it's basically a root mean square difference between the positions of a bunch of atoms. A more sophisticated one would be to define an energy function between the molecule that I'm designing and the molecule I'm trying to bind to. And David Baker, University of Washington, has written this software called Rosetta, and that they can use to computationally design protein interfaces that bind other proteins. They've been very successful at that. I want to use the same kind of idea here. So you define an energy function, a scoring function, and you dock it against the protein. And then you mutate this and you dock it again. It's a more expensive computational process. But that's what I think Monte Carlo, I mean, he uses Monte Carlo. He's the one who taught me that. He said, that's the way we're going to solve these problems. It's Monte Carlo. Yes?
We had a very interesting analogy between molecular design and software. Software design, physical, subdivisions possible. So you can put those. Is there anything analogous in the molecular realm? Like, I noticed you didn't mention vector nanosystems. You can't build any of those things, of course, but if you could build them, then you could, like, do machines, like cars made out of parts and subparts. You don't have to search.
I want to build things like that. And in 2 days, Eric Drexler and I will be at a Department of Energy workshop talking about that. I hope I'm allowed to say that. Yeah, it should be. It's DOE. Yeah, and atomically precise manufacturing. I think for the sort of intermediate, building bigger bricks that we could then connect together is my goal here for that part of it. So if I can build, I think we're going to need to use Monte Carlo to design things like this. But if I can design a couple of bricks where I can put functional groups on the outside, I could link up with other bricks, and I can then I could manhandle one on top of another and snap them together potentially to make bigger, atomically precise materials. Then I've got something like a subroutine that I can connect to another subroutine to make something that's more than the sum of its parts. Yeah. Yes?
I have a question about garbage collection. Did you write your own garbage collector?
No, I'm using the memory pool. I'm using Boehm to bootstrap, and I'm using the memory pool system by Ravenbrook, which is a C library that does copying garbage collection. It was developed at Apple for Dylan, this language that they were developing for a while, and it is it's a wonderful, complex garbage collector that does moving garbage collection. It's written in C, and I've just hooked it into my thing. The thing about it is you have to, when you move a C++ object from one place in memory to another, which people, in the C++ chat room told me was impossible, you have to update every pointer that points, the pointer here to there now when you moved it, right? And if one pointer gets out of place, that thing will tear itself apart. The program will crash. So I wrote a static analyzer in Common Lisp that uses AST and AST matcher library, and it finds every pointer and writes about 20,000 lines of C++ code that updates every pointer continuously. And it runs, it works perfectly.
You have pauses.
So that garbage collector is a generation, it's a generational garbage collector, and they've worked hard to reduce the pauses. I'm not so worried about it because scientific programming doesn't really need, I just want it to run as fast as possible and clean up my garbage and work in a fixed memory footprint. Because if I run this on supercomputers where I've got 2 gigabytes to work with, I can't have a garbage collector like Boehm, which will fragment and eventually run out of memory. It makes me very uneasy. So also it's a generational garbage collector, so it continually compacts memory, and it runs faster than Boehm by about 2-fold in my timing of it a couple of months ago. Yes?
You mentioned looking at Steam Common Lisp. Did you consider?
Back then, it wasn't a thing. And no, I didn't want to work with Java and the Java sort of infrastructure. LLVM looked much more like what I wanted. And it was a great decision. I love that whole infrastructure. The software is wonderful. The libraries are wonderful. Things really well thought out.
All right. Thank you very much, Christian. And I think you'll be here for a few more minutes to answer like kind of offline questions. But thanks for coming to Google and talking about LLVM and CANDO.
Thanks for inviting me.
Yeah.